Welcome everyone. Welcome. Hope everyone is doing well today. So today we're going to be diving into a brand new topic. We're going to be looking at macromolecules and membranes. The former is not so exciting, but I find membranes to be particularly very exciting as a biological topic. So I don't know if that means anything to you guys, but I find them exciting. And so before I get started, I just wanted to just share my screen. Well, I'm gonna share my screen anyways. Um, and just to highlight some things about what to do. So there's a slides for today. There's another extra credit discussion board. I saw a couple of you have already done it. Good job. And then your textbook chapters are here, and your measurements and statistics laboratory report is due this week. It's pretty simple. It tells you everything you need at the end.
Professor,
Yeah.
the due date passed. It was Monday.
Okay. Well, it was due this week.
Yeah.
It was due this week. So, yeah. What's your question?
So I just want to say that the homework too, that we already did it. So this ones we can also cannot see the things we got wrong and right. So
You
I cannot see what's right or wrong too. Maybe see what I got wrong.
you should be able to. Test options. You should be able to. All answers after the due date. Yeah, this should be available for you to see. So all answers should be available for you to see. So you have to go back and click the assignment and view your assignment, but they are available for you to see.
Okay, I will try one more time. So
Yeah. I mean, that's the only option I have to make grades available to you. So if that doesn't do it, then they would require me to like literally print out your thing and send it to you. So, and I'd be printed out as like make a PDF of it. So. And then you have your lab for this week. So you're going to be hopping on over to Labster, and you're doing an exercise on introduction to food and macromolecules and how your own nutrition is influenced by what we're going to talk about today. So it's going to be it's an interesting lab. You analyze the content of food and you think about how these macromolecules, which you're going to talk about, is related to your own nutrition. So I figured that's something everyone would like. So so in terms of today, we're going to, as mentioned, we're going to talk about macromolecules and membranes. If you have any questions throughout, you can dump it in the chat. You can raise your hand, or you can just talk out like the people that have done thus far. It is up to you. We're not going to have any exercise today. We will next week, but this week, because we got a little bit behind because I was sick, so I just want to catch up on this day. So we're not behind. So again, any questions, please let me know. It could be questions about your grades or about the materials, just let me know. Now, macromolecules are simply just large molecules that are comprised of thousands or potentially several connected atoms. And when we're thinking about a macromolecule in the context of biology, we're thinking about proteins, carbohydrates, lipids, and our nucleic acids. But they start with and are pretty much comprised solely of the most amazing element on the planet, which is our friend carbon. Now, as we discussed in the chemistry lecture, carbon is a really amazing molecule. It is the most important thing for life. It is the second most abundant thing in your body. But carbon provides this really important framework of biology because carbon always binds to four things. It never binds less, never binds to any more. And because of carbon's properties, it likes to bind with other things, in particular things that are associated with life, including oxygen, nitrogen, sulfur, phosphorus, and hydrogen. And in addition to bonding to all of these, we can have what are called hydrocarbons. And so the way you break this down is hydro meaning hydrogen and carbon meaning carbon. And so you'll see hydrocarbons decent a bit in sort of part of this lecture, but these are molecules consisting only of carbon and hydrogen. And these are also non-polar. I will also mention one thing is hydrocarbons are what make up oils and fuels. So what you fill your car up with is a hydrocarbon. Now, when we start to break down what attaches to carbon, we have what are called functional groups. And these functional groups are specific molecular groups that bind to carbon and hydrogen cores. And as you'll see, there's a number of groups that are important for biology, but each of them has their own chemical properties. And these properties of these groups, these functional groups, are retained when they attach to anything. So it could be a carbon, could be anything, and they influence the behavior of the molecule and the reactions. And so the most common groups you're going to see in this class, and you're going to see going forward, are hydroxyl groups, so OHs. So in this case, we have ethanol. So we have carbon bound to just a hydroxyl group. So if you like to, well, you guys are probably too young, but if you like to drink beer and wine, this is what you're drinking. Ethanol is what makes beer and wine have their, alcohol flavor. Then we have carbonyl groups, which is a C double bonded to an O. And we have carboxyl groups, which are pretty important. We'll talk about carboxyl groups a lot when we start to talk about proteins. We have amino groups, which are NH2, sulfur hydro groups, which are SH, phosphate groups, which are PO4, and methyl groups, which are CH3. So these are our primary functional groups. And each one of these has very different properties. They're found in different types of these large macromolecules. So some are found in carbohydrates, some are found in proteins, some are found in your DNA. But these are the most important ones. You don't need to remember all of these. You might, as you sort of might start to remember, OH is hydroxyl, but you don't need to remember all of them. Just to know that these are very important functional groups, and each of them has different properties. Now, another aspect of thinking about macromolecules is what we call an isomer. And molecules that are isomers have the same molecular formula, or what we call the empirical formula, and they differ in how they're grouped. And so they can be what are called structural isomers, and they differ in the structure of, say, their carbon skeleton, or they can be called what we call stereoisomers, which differ in how these groups are attached. And I can just show you what I mean by this. In particular, we're going to look at enantiomers, which are essentially left and right-handed parts of anything. And I always find this one to be kind of weird, to be perfectly honest with you. Left-handed, right-handedness of chemical atoms, but that's really what it is. We have a central carbon bound to X, Y, Z, and W, right? The left-handed version here is oriented like this, but we could do a mirror version of that, and we have the D version of this, which is weird, right? Same atom, same chemical formula, but different orientation in 3D space. So this is what we mean by an isomer. You can arrange the atoms in different ways, have the same chemical formula, but the interesting thing is if this molecule here on the left and this molecule on the right, they behave very differently. And it's just kind of a weird thing about chemistry. And this is the part of chemistry why I've never particularly liked, but just to know you can have isomers and it's the same chemical formula, slightly different shape. And as you will learn throughout this class, the shape of any molecule, the shape of any protein, the shape of any macromolecule will dictate its function. And being, left-handed versus right-handed, going from a chain to a ring, it all changes your shape. And when you change your shape, you change the potential function of your molecule. Because again, this molecule on the left here, that's left-handed, versus this one on the right, which is right-handed, they have a different shape and they'll function slightly differently, even though they have the exact same chemical formula, which I think is weird, but it is what it is. Now, macromolecules are comprised of essentially two main components, polymers and monomers. Now, monomers are just very small, similar chemical substrates, or subunits, I should say. And as you string multiple monomers together, you start to get what are called polymers. And so what I mean by that? Well, we can look at the chemical structure of a carbohydrate. So in this case, we have starch inside the chloroplast of a plant. The monomer version of our starch is a monosaccharide or a sugar. But as we attach a bunch of these monosaccharides or sugars together, we form what's called a polymer, and that polymer has a name called starch. So that's an example of a carbohydrate. But we can also look at something like DNA or nucleic acid. Our monomer here is our nitrogenous base. So ATG are attached to a sugar and attached to a phosphate group. That's the monomer. Now, as we link those monomers together, we start to create strands of DNA. So as we link A, T, G, and C together, we create strands of our nucleic acids. And at the sort of the higher level, you have structural parts of the cell like a chloroplast or a chromosome. But it all starts with a monomer linked together to form polymers. And so this is an important distinction because for carbohydrates, for proteins, for nucleic acid, you start with a monomer link them together with some form of bond, then you form a polymer. And that polymer is basically the building blocks of what we are talking about, right? It's our proteins, it's our complex carbohydrates, it's our DNA, it's our RNA. But again, it all starts with monomers being linked together to form polymers. And just again, just to sort of show you here, for proteins, we start with an amino acid, which is the basic monomer of a protein. We link a bunch of them together. We form a polymer, which we would call a polypeptide, and that leads us to proteins, such as, in this case, we have what's called a filament. Now, it's not quite the same for lipids. Lipids, there are no monomers. You don't link a bunch of things together with lipids. But when you start to look at lipids, you can take one lipid structure and bind it to a couple others to form more advanced structures. But again, it's not really a monomer polymer here. Lipids don't really follow that sort of structural formula like your DNA does, like your proteins do, like your sugars do. Now, in terms of how we assemble our polymers, again, it all starts with monomers, and we have to link them together. Now, typically, the way we are linking our monomers together is by what we call dehydration synthesis, and we have dehydration synthesis here on the bottom. And what you're seeing, we're joining this monomer here to this monomer here. And what we're doing is to join them, we're essentially creating water. We're taking water out of these molecules. So you're seeing we're getting rid of OH and an H here, producing water in the process, which is why this is called a dehydration synthesis, because we're synthesizing a larger molecule from two smaller ones, but to do it, we're removing water from those original starting molecules. So that's why it's dehydration synthesis. Now, this is in contrast to hydrolysis, which is how we break down polymers into monomers again. So we have some sort of polymer here, and to break that polymer apart into its monomers, we have to add water, which is why we call it a hydrolysis or a hydraulic reaction. This is how we form, and this is how we break our monomers and polymers and back and forth. Now, start out talking about one class of our macromolecules. These are our carbohydrates. And our carbohydrates typically come with a ratio of 1 to 2 to 1. And this is a ratio between carbon, hydrogen, and oxygen. Now, you'll see this empirical formula, CH2, and then this little subscript N. And this is a basic formula for a carbohydrate. Anytime you see CH2O like this, it's referring to some sort of carbohydrate. So for shorthand in a lot of biology textbooks, they'll write CHO like this, just as a note. Now, the thing about carbohydrates is these carbon-hydrogen bonds hold a lot of energy. And as you break those bonds inside your cells, you release energy. And because breaking bonds releases energy, carbohydrates are fantastic molecules getting energy from. They're also fantastic for storing energy. And you've definitely heard of many of these types. Carbohydrates are sugars, they're starches in plants, and it could also be something as simple as glucose, which is sort of the most basic of all the sugars. Now, the building blocks of carbohydrates start off as monosaccharides or monomers. And so a monosaccharide, again, it's just a monomer, and it's the simplest type of carbohydrate out there. It's a sugar with 6 carbons, and that these 6 carbons ultimately play, and these 6 carbon compounds ultimately play an important role in metabolism. So the easiest example I can give you of this is glucose. C6H12O6, right? So it has that CHO formula, that backbone for carbohydrates. But again, this is a very simple carbohydrate. And glucose as a compound has both a structural isomer and a stereoisomer. Fructose and galactose, they, again, same chemical formula, slightly different arrangement of the molecules. And there's actually sort of an interesting thing is when you look at the metabolism of glucose or fructose or galactose, enzymes actually act differently upon them. So when we talk about enzymes, this will become more clear what I mean by this. But just to know, like, just even though they have the same chemical formula, the way your machinery of your cells acts upon them is actually very, very different. Again, it's all reflected based upon that structure. And so I can just sort of show you what I mean by this. We can have a linear form of glucose right here. And then we have the what we call the ring to the circular form of glucose here. And we can change slightly the orientation of the OH. So the OH is here for beta glucose, and the OH is here for alpha glucose, but these are two very different molecules. They behave differently. Same chemical formula, same shape, but how you move this hydroxyl group, either up or down, makes one glucose and the other one galactose. And then we have our fun friend fructose over here. Again, same chemical formula, slight differentiation in how the molecules are attached and how they're oriented to themselves in 3D space. And we just talked about this, so we'll move on. Now, as we start to link monosaccharides together, we can form what's called a disaccharide. So when I link two monosaccharides together via dehydration synthesis, so again, and as you can see from here, I'm going glucose plus fructose, and I'm removing water from both of the molecules, creating a disaccharide called sucrose, aka what you put on your oatmeal or what you put in your coffee or your tea, right? It's just the sugar you use in your everyday life. Now, disaccharides aren't much different than monosaccharides. They just used typically for storage of energy, and they can also be used for transport as well. And there are a number of different types of disaccharides out there. I mentioned glucose. For those of you that like malted milk balls, I think those are gross, but to each their own. Maltose, the sugar, is what gives malted milk balls their sort of malty flavor, whatever you want to call that, whatever that flavor they have. And then also something like lactose. So for those of you that like to drink milk, dairy-based milk, that is not like a plant protein. Now, after our disaccharides, we move up to the sort of the most complex form of carbohydrates, which we call polysaccharides. And these are long chains of monosaccharides, 3 or more monosaccharides linked together. Very much like a disaccharide, polysaccharides are made through dehydration synthesis. And polysaccharides are typically used for long-term storage of energy. Because remember, the CH bonds inside of a saccharide contain energy. And as you break those bonds, you liberate energy for your cells. Now, energy storage is commonly what we see polysaccharides used for. We see starch in plants, and as animals, we use glycogen. So when you eat, say, like a nice big Chipotle burrito or a big pizza or something like that, you can't absorb all that energy at once. So what your body does, it creates a polysaccharide called glycogen for short-term storage of your energy. Long-term storage of energy in animals like us is obviously fat, right? But we'll get to fats later in this lecture. But in addition to energy, they actually offer structural support. So for instance, plants use cellulose as a structural support of their cells. And so we can look at our formation of our different types. So we have glycogen, which should be found in animals. So you can see again, it's just a long chain of monosaccharides linked together. And the same thing with our starch. It's all just long chains of monosaccharides linked together. That's it. So polysaccharides, just long chains of monosaccharides. So it's kind of nice. It sort of builds upon one another. You start with a simple sugar and you start linking together disaccharides and eventually link them together long enough to create polysaccharides. Now, I mentioned cellulose. Cellulose is a particularly important, the most abundant macromolecule on the planet. It accounts for about 15% of all the biomass on the planet. And what do I mean by biomass? I mean just living tissue. So 15% of all living tissue on this planet, the mass of it is cellulose because plants are so abundant. But cellulose is a really stable compound. And it forms not only these sort of long strands, but it also forms vertical linkages that give plants that have cellulose very, very strong cellular structures. So our polysaccharide here, again, it's not just for energy, it's also used to support structures of plants. So one of the reasons why trees can go very, very tall is because of this of the structural support of our friend here, cellulose. Very, very strong. So that's our sugars. Next up is going to be our nucleic acid. And this includes DNA and RNA. Now, our basic sort of building block of DNA and RNA is our monomers, which we'd call our nucleotides. And each nucleotide, as I'm sure you've probably heard, is comprised of a sugar, a phosphate, and a nitrogenous base. When we're looking at the different types of nucleic acids, whether it's DNA or RNA, the sugar in DNA is deoxyribose, and the sugar in RNA is ribose. So they are very, very similar, and I'll show you what that looks like in just a moment, but they do differ slightly. Now, the nitrogenous base parts include purines, which include adenine and guanine, and our pyrimidines are thymine, cytosine, and uracil. And our nucleotides themselves are linked together by what we call phosphodiester bonds. You don't need to memorize everything, but I just wanted to put that information out there. So let's look at this structure. We have the nitrogen space here, which would be A, T, G, or C. Oops. It's linked to a five carbon sugar. In DNA, remember DNA is deoxy, means it's missing this oxygen right here. RNA is just ribose, so it has the extra oxygen here. So very subtle differences in the sugar, but they give RNA and DNA very different properties. And then finally, this 5-carbon sugar is then connected to what we call a phosphate group. Remember, phosphate groups are just PO4s. This is something that you'll want to remember what a phosphate group looks like, because phosphate groups are going to come up a lot during this semester, especially when we start to talk about energy. Now we can start to look at the structure of our different nucleic acids. So remember, our nucleic acids are our monomers. Now, as I mentioned, we have purines and pyrimidines. Now, purines include adenine and guanine, and you can see they are much larger than our pyrimidines here at the bottom, which include cytosine, thymine, and uracil. Now, before we go on, I just want to mention that for DNA, we use these 4 bases here, A, T, G, and C. But for RNA, we swap out thymine for uracil. So for RNA, we have A, G, C, and U. For DNA, we have A, G, C, and T, just as a note. But ultimately, we could look at how these nucleic acids are linked together. So we have our nucleic acid here with 5-carbon sugar, and that 5-carbon sugar, remember, is bound to a phosphate group. We link our nucleic acids together via their phosphate to the carbon here by what's called the phosphodiester bond. And this is just, again, the monomer is here, and we just link the monomers together to form polymers. And the polymers are just nucleic acids, DNA, and RNA. Now, why are we talking about DNA? Well, as I'm sure you probably know, DNA is kind of the bee's knees of the cell. It basically tells everything that there is about you in just a sequence of DNA bases. So everything that you can do as a person is encoded in your DNA. It is the way you inherit DNA from genetic information from your parents. It's how you pass genetic information on to the next generation as well. So DNA, as I said, it's the bee's knees. It encodes everything about you, all the traits that you have, height, eye color, so on and so forth, encoded in your DNA. And it doesn't just encode big things, it encodes small things. So the proteins that you can make, all encoded in your DNA. And the way to encode it is just by changing the order of A's, T's, and C's. That's really all it is. If you have a string of A, T's, G's, and C's in one order, and then it's in a different order, that's different DNA, which encodes slightly different instructions. So it's pretty simple, which I think is nice. Cellular things at this level can be particularly very simple, which is handy for learning, but it's also sort of handy for life as a whole. Now, DNA has a overall, we would call a secondary structure, and this is what we call the double helix. And the double helix refers to this idea where we have 2 strands of DNA connected together by hydrogen bonds. And this leads us to the base pair ruling, and you're going to want to remember this. A always pairs with T in DNA, and C always pairs with G in DNA. Now, in RNA, A always pairs with U, but C still pairs with G in RNA, just as a note. So A with T, B with C, or A with U. Very important rules to remember. Very, very important rules to remember. Simple rules, but important. Now, we can look at the structure of DNA. Again, it's a double helix. It looks like just you took a ladder and you linked it to, right? So each one of these backbone, each one of the DNA connecting, the nucleotides connecting, is the rungs of the ladder. And that's all DNA really kind of looks like. It's just like a ladder that you then twist. And that twist is what is the double helix. But again, it all started off with monomers. So we have the nucleotides here, the sugar, the phosphate, and the nitrogenous base here, linking them together over and over again. And that's how we form the structure of DNA. Now, I mentioned that the DNA molecules are held together by hydrogen bonds, but actually some brand new research has actually showed that's not the case. So if you want to read about this really brand new research, I would take a peek at this. But for the purposes of this class, we can still consider the DNA being held together by hydrogen bonds. But again, if you want to know what cutting edge research says, take a peek at this link right here. So RNA, as I mentioned, is very similar to DNA except for the nucleotides, as well as the sugars, right? So RNA has ribose sugar instead of deoxyribose sugar, and RNA has uracil instead of thymine. And RNA, unlike DNA, is a single nucleotide strand. But ultimately, all RNAs, and we'll talk about this much more in depth later this semester, is just a copy of a DNA strand, because RNA is used as a template to make a protein. And again, we'll talk about that as the semester progresses. But we can just do a comparison of what they look like. The same sort of basic structure. They have that one linear strand where you link your sugars to your phosphate groups to form a backbone, and then you have your nitrogenous bases here. DNA just has two strands. RNA has one, and DNA has thymine, or your RNA has uracil. And again, the only other major difference here is the type of sugar. Ribose sugar for RNA and deoxyribose sugar for DNA. And so before we sort of go on to talking about proteins, we want to talk about a very specific type of what we of a energy molecule, which we call ATP, or adenine triphosphate. Now, ATP is the energy currency of the cell. And ATP looks a little bit like this. It's a 3 phosphate groups to a 5 carbon sugar and a nitrogenous base. In this case, that nitrogen adenine. So it sort of straddles this line between being a sugar and being a DNA base at the same time. But you don't need to sort of memorize the structure of this yet, but I just wanted to put it forth because when we talk about cellular respiration, ATP is going to become very, very important because it is the energy currency of your cell. If you eat a Chipotle burrito bowl or a pizza or something like that, your cells convert the energy stored in that pizza or that burrito bowl into ATP to do all the functions inside your cell. So keep this in the back of your mind for later in the semester. Next up in our macromolecule train is going to be proteins. And proteins are basically the workhorses of the cell. They do pretty much everything. Every cellular process that we're going to talk about this semester, we're going to talk about a protein in some shape or form because proteins act as catalysts for chemical reactions. They act as defense for your cells. They're involved with transport, support, motion, regulation, and storage. They do everything. Every topic discussed this semester involves protein in some shape or form. So we're not going to talk too in depth about proteins right now. We'll talk about the basics of them, but we're going to talk more about them later in the semester. And pretty much every topic we discuss, we'll talk about proteins. Now, proteins as a sort of a big macromolecule are polymers. And these are made up of our monomers. And our monomers, in terms of a protein, are what we call amino acids. And amino acids have a very conserved structure across all of them, and I'll show you a table of them very shortly. But all amino acids have the same basic structure. They have a central carbon bound to a hydrogen, and that central carbon is then bound to an amino group, NH2. And that central carbon is also bound to what we call a carboxyl group, or COOH. Now, all amino acids have this exact same, what we call the backbone. This carbon to the nitrogen, to the hydrogen, to the COOH. Now, where each amino acid differs is in this R group, or what we sometimes call a side chain. So let's look at the 20 amino acids that we see here. So we have all proteins are made up of the exact same 20 amino acids. And the amino acids, as you can see from this table, have the same backbone. That COOH, right? This backbone here. It's the same backbone here, same backbone here, and so on and so forth. So there's no variability on that backbone. But where each one differs is on the side chain or this R group. So we can look at something like alanine here. It has a side chain of CH3. That side chain is very different from something like glutamic acid, which has this very large and also charged amino acid side chain. So each of the amino acids has different R groups, but the same backbone. And these different R groups give them very different functions. And so there are 4 sort of main functional groups of our amino acids. These include those that are nonpolar, polar that are uncharged, charged either positively or negatively, or what we call special cases. These ones down here that says aromatic, these are also polar. These ones are also nonpolar. Um, the aromatic ones are very, very hydrophobic, and that will come up later in the semester, but just as a note. It falls into one of four categories: nonpolar, polar, charged, or these special cases, which include proline, methionine, and cysteine, or cysteine, depending on how you want to say it. All have that same backbone here, same exact one, but what differs is this R group, and the R group gives each one of them their distinct properties. And we'll talk about what that means very, very shortly in the lecture as we talk about protein structures. But keep that in the back of your mind. Now, how do we join proteins together? Well, proteins are joined together. Those monomers are joined into polymers via what we call dehydration synthesis. And the special type of bond that forms polymers of amino acids is called a peptide bond. And the way protein is combining amino acid 1 with amino acid 2, again, via this peptide bond. Again, we're producing a water, so it's dehydration synthesis. But the way this bond works is we link we have that carboxyl group, that COO, binds to the amino group in another amino acid. So we have the production of water. So HHO produces the water. And so we end up binding this amino acid to this amino acid, forming this, what we call a dipeptide, or two amino acids linked together. But again, we produce her, but we link them together on their backbones. And as you can see, the R groups are not involved here, right? So this R group can be variable, right? But it's not involved with how we join amino acids together, because remember, as we join amino acids together, more and more of them, we make proteins. And proteins have, as you start to link all the amino acids together, they have 4 levels of structure. And the 4 levels of structures of proteins start with the primary structure. And this is just simply, what is the sequence of amino acids? That's it. How do I link those 20 amino acids together? That's all the primary structure is. Now, the secondary structure is starting to get a little more complicated. And this comes in 2 forms. We can either have what's called an alpha helix, which is a coiled spiral, or a beta sheet, which is a planar structure. And I'll show you what that means in a second. But keep that in the back of your mind. The tertiary structure is essentially the final folded shape of a globular protein. And this is stabilized by a number of forces, but this is typically the final level of protein structures. So if you make a protein to do, say, break down lactose, that protein's final structure to break down lactose will be its tertiary form. And then finally, we have what's called the quaternary form. It's when we arrange multiple proteins together to form large globular units. And I'll show you what these mean right now. Now, so again, the primary structure is just a sequence of amino acids. So how do I link any amino acids together? Now, the secondary structure forms either beta sheet or alpha helices. And beta sheets look very much just like this. They look like you basically took a piece of paper and folded it, and then you unfold it and you have the sort of the kinks on the paper. That's what a beta sheet looks like. And an alpha helix looks very much like your DNA. It kind of twists into a helix. And these are held together by different types of bonds. But as you start to link together beta sheets and alpha helices, you start to form that tertiary structure. So as you can see in this globular protein, we have beta sheets connected to alpha helix. We have beta sheets connected to more beta sheets, then connected to alpha helices, and then beta sheets and alpha helices. That's all really a protein's tertiary structure is, the 3D orientation of these structures together. And then finally, we can have what's called the quaternary structure, where we take multiple globules of protein and interact them to form a megaprotein, essentially. So it has like an overall specialized function. We won't talk too much about the quaternary structure. We're mostly going to focus in this course on these 3 primary levels: primary, secondary, and tertiary. And there are some other other structural characteristics of proteins that I'll just mention. These include motifs, which are common elements of secondary structure, many proteins that are useful for determining the function of unknown proteins. And then we have what are called protein domains, which are functional units within the larger sort of quaternary structure. We're not going to talk about those, but I just wanted to make note of these because you might see them in your textbook or readings or something like that. And I just wanted to put that information out there for you. And this is just a sort of just sort of a depiction of the motifs and domains of proteins and what it looks like. But we can also think about what happens when we denature a protein. And as I mentioned, proteins have a three-dimensional structure, in particular in that tertiary form. So that sort of final level of structure that we care about. And when a protein is in that final level of structure, if you denature it, you change its shape. And it typically goes from, you have that sort of like globular protein, and you change its shape, causing it to unfold back into its primary amino acid sequence. That's what we mean by denaturation. When a protein loses its shape, and as I mentioned, if you lose the shape of your protein, or you lose the shape of anything in terms of our macromolecules, you change the function. And so denaturation occurs via a number of mechanisms, including changes in the pH, changes of temperatures, or the potential ionic concentrations of a solution. You can also denature your own proteins if you wanted to. So you could actually take the urea in your urine and denature a protein with it. So just as a, if you ever wanted to know, what your urine's good for, it's good for denaturing proteins. So fun facts. But again, to show you what this looks like in real life, we have a properly folded protein here. It undergoes denaturation by, say, increasing the temperature, and it causes the protein to unfold into a or sort of unfolded protein. And that's all we mean by denaturation of proteins. It's just causing it to lose its shape. And so the final category of macromolecules we're going to discuss today are the lipids. And this is sort of a loosely defined group of molecules with one main chemical characteristic. And all lipids themselves are insoluble in water. And what do I mean by insoluble in water? Well, it means they just can't dissolve in water. So I'm sure you guys have all seen this. If you take, say, vegetable oil, which is a lipid, and you pour it into a pot of water, you'll notice that the oil separates from the water, right? And it separates from the water because the lipids are not soluble in water. And that's a characteristic all lipids share. And all lipids have a lot of things in common, including a very high proportion of carbon and hydrogen bonds, which causes them to be very, very hydrophobic, meaning they fear water. And there are many, many types of lipids: fats, oils, steroids, waxes, vitamins. There's all sorts of different classes of lipids. But again, they all share things in common. They're typically hydrophobic and they're insoluble in water. And they have a very high proportion of carbon and hydrogen bonds. So let's talk about a couple of these lipids, and we'll start with fats. And the first fat we'll talk about is triglycerides. And so you guys have probably heard at some point what a triglyceride is, but it is a triglyceride is simply one glycerol bound to three fatty acids, and I'll show you a picture of that very shortly. So that's triglycerides. And then we have fatty acids, and fatty acids, these are just, as we'll talk about, are just strings of carbons and hydrogens linked together. And these fatty acids can be variable in size, they can be variable in length, they can be variable in what their chains actually look like. And again, I'll show you that in a second. And they can be unsaturated or saturated or transsaturated. And you guys have probably heard of saturated, unsaturated, and trans fat. And what do I mean by those things? Well, saturated fats have no double bonds. Unsaturated fats have one or more double bonds, and trans fats are not natural compounds, but they're actually produced industrially. And I'll show you what all three of these look like in a second. But our saturated fats have a much higher melting point. They're animal in origin. Our unsaturated fats have low melting points, and they're typically plant in origin. So let's look at some fats. Let's look at some fats because that's, it's exciting, right? So we have two fats here. One here that's saturated, so you can see there's no double bonds present. And then we have unsaturated ones where you can see there's a number of double bonds present here. And they have overall similar structures, right? They're chock full of carbons and hydrogens, and they have some oxygens here at the top as well. And what you'll notice is that the structural formula looks very different. So when we look at a 3 model, our saturated fats have very straight fatty acid chains. So this is a fatty acid chain here. And our unsaturated have slight kinks in their chains. And their kinks are due to the presence of these double bonds. So in terms of your overall health, typically speaking, saturated fats are healthier and unsaturated fats are not as healthy. And one of the reasons unsaturated fats are not as actually healthy is this kink makes them get stuck. They're sticky. And so it's not particularly good for your blood vessels when you have sticky fats getting stuck in them, or potentially stuck in your heart, or potentially stuck in your gut. So health-wise, your unsaturated ones, not so good. Saturated fats are typically very good. But in terms of health, it's usually about quantity as opposed to what you are eating. Just as a note. Now, in terms of biology, one of the more important classes of lipids are going to be our phospholipids. And this is actually transition to the second part of the lecture where we talk about membranes. Now, our phospholipids are comprised of glycerol, two fatty acid chains, and a phosphate group. And these are what form all biological membranes, whether it's a bacteria, whether it's an archaea, whether it's a eukaryote, whether it's you, it doesn't matter. All membranes are formed by phospholipids. So let's look at what a phospholipid looks like. So in terms of what we're looking at, we have a polar and hydrophilic head, meaning the heads of these, which are depicted like this typically in drawings. So when you look at your textbook, you'll see phospholipids typically depicted like this. Their heads are hydrophilic, meaning that they love water. However, their tails are comprised of fatty acids, which means they are very hydrophobic. But the basic structure looks a little bit like this. We have a choline bound to a phosphate group, bound to glycerol, and that glycerol has two fatty acids attached to it. Fatty acid chain 1 and fatty acid chain 2. But what you'll notice here is that fatty acid chain 2 here on the right has a double bond, so it's not fully saturated, unlike the chain here, which is fully saturated. And so all all phospholipids have typically one chain that is saturated and one chain that is unsaturated, which gives the unsaturated chain that slight kink. So anytime you see that slight, this overall structure, that slight kink is again due to the presence of that double bond, which changes the structure. This is what all phospholipids look like. Bacteria, archaea, us, basic structure. So it'll behoove you to remember this basic structure. Not so much the choline part, but mostly the phosphate bound to glycerol, bound to two fatty acid chains. That's the sort of the meat and bones of a phospholipid. Because phospho is phosphorus, lipid is fatty acid chains. So that's the basic structure of our phospholipids. Now, because of the properties of the phospholipids, a hydrophilic head and a hydrophobic tail, they actually form naturally what we call a phospholipid bilayer. So if you took your phospholipid in one of your cells, broke it apart, and threw it into water, it would spontaneously actually form this bilayer shape. It would form a sandwich-like shape. And the way it orders itself is it takes the hydrophilic heads and puts them close to the water, thus shielding the very hydrophobic tails from the water. So all phospholipids form bilayers like this. This overall sort of like sandwich-looking structure. Again, it's all due just to the properties of the phospholipids. Hydrophilic heads, hydrophobic tails, causes them to form in such a way where the hydrophilic heads are exposed to the water and the hydrophobic tails are hidden away from the water. And you might think, well, why does it matter with water? And just as a note, all life it has to be surrounded by water, right? Life can't exist if water is not present, and your cells are basically just big bags of water, and what's inside your skin is just a big bag of water. So and even when you have things are growing in nature, again, they're still just bags of water. So everything is all about water. Just as a note. And so the bilayers form like this, again, all based upon the properties. And these bilayers are going to be a nice transition to talking about the next part of the lecture, which is going to be focused on membranes. Now, as I mentioned, our membrane structures are all based upon phospholipids. And these phospholipids, as we mentioned, are arranged in bilayers. But in addition to the phospholipids, you can embed a number of different things inside those membranes, including proteins, other types of fatty acids, steroids included, cholesterol, anything you kind of think of, you can embed inside that membrane. And we'll get to those as the lecture progresses. But before we sort of go on and talk about membranes, we need to talk about this concept of what we call the fluid mosaic model. And the idea being is the fluid mosaic model really describes this idea of basically your phospholipid bilayers, your membranes, being fluid. They're not static, right? They're not, like a solid surface like my water bottle here, right? What makes up your cell isn't solid, it's fluid. It's constantly in motion. So as we mentioned, our membranes are made up of these phospholipids. So just as a refresher from 5 minutes ago, hydrophobic head of phosphorus and glycerol attached to 2 fatty acid chains. Now, this fluid mosaic model of cell membranes is kind of interesting. Now, as I mentioned, your cellular membranes are not, they're not like concrete, right? They're not a solid surface like my water bottle here. These phospholipids are moving constantly. And that's kind of a weird thing to think about. What keeps your cells together is kind of always moving. But it's also fluid in the sense that you can embed stuff in them, including proteins, cholesterols, different types of sugars. And so they're not just all phospholipids, it's fluid, it's moving, and there are other things embedded inside your membranes, giving them different properties as well as different functionalities. But our cellular membranes themselves have 4 key components. The phospholipid bilayer, they have what we call transmembrane proteins. So these are proteins that that typically span the entire membrane. So in this example here, this protein would be transmembrane because it spans the whole membrane. We have interior protein networks, so they're peripheral or intercellular membrane proteins. And then finally, we have what are called cell surface markers, which include glycoproteins as well as glycolipids. So as we mentioned, our lipids are the backbones of our membranes. They're comprised of glycerol in the 3 carbon chains and so on and so forth. And they do spontaneously form a bilayer themselves. And as I mentioned, these bilayers are fluid. Now, what this sort of looks like, and when you sort of visualize it 3D, is they moving constantly either laterally, so we're moving left to right or top to bottom in this diagram here at the bottom. And sometimes they actually will flip-flop between sides. So you have an exchange of one phospholipid that's on this side with a phospholipid on the other side. But they are constantly in motion. They're fluid. Again, it's a weird thing to think about because a membrane is a thing that separates outside from in. But your membranes have to physically be able to move because as we'll talk about, if they were completely solid, you couldn't move stuff in and out of your cell. And if you couldn't move stuff in and out of your cells would die, right? They would starve or they would poison themselves with whatever was already inside them. So they have to physically be able to move. And for them, these phospholipids to move, it gives them fluidity and allows them to transport things across your membrane. Because again, if you have a solid structure, like my water bottle, I can't move things directly through this water bottle because it's not fluid, it's solid. But my membranes are not like my water bottle. They're fluid. They have the capacity to allow things to move through them. And that's an important feature of membranes. They need to be tough, but they can't be too tough. Because again, you need to move stuff across those membranes. Now, in terms of how fluid these membranes are, again, just the movement of these, you can influence how much your fatty acids move by doing a number of structural changes to your membranes. So as you saturate your fatty acids, the membranes themselves become less fluid than unsaturated fatty acids. In addition, warmer temperatures make membranes more fluid than colder temperatures. So membranes that are at higher temperatures are moving faster. So those phospholipids are moving faster up and down, back and forth, side to side, much faster than if they were at a colder temperature. And you can also change the lipid composition. And so you can change what types of proteins. So you can go from a bilayer to a monolayer. I'm sorry, not proteins, lipids. You can go from a bilayer to a monolayer, and that will affect fluidity as well. So you can change the properties of your fatty acids, your phospholipids here, to influence how fluid they are. And it all depends on the environment. Because again, living in a cold environment requires different adaptations of your membranes than living in a warm environment, or potentially living in an acidic environment versus a neutral environment or a basic environment. So membranes aren't static. They can be changed not only sort of on small scales, like within an hour or so, but they can also be changed permanently to make them, say, better suited for a different environmental condition. So as I mentioned, we can embed membrane proteins. Proteins into our membranes, and there are a number of classifications of our proteins, including transporters. And as you can see from this diagram, transporters move things from inside the cell to outside the cell, or outside the cell to inside the cell. We have enzymes, which catalyze chemical reactions. We have proteins that can act as cell surface receptors, so something binds to them and gives a signal to the inside of your cell. We can have proteins that are involved in cell with a cellular identity. So for instance, on your liver cells, there's a special type of protein that has a sugar attached to it that says, hey, I'm a liver cell. And the same thing for your heart, the same thing for your lungs, the same thing for your muscles. There are also proteins in your membranes that are involved in cell-to-cell adhesion, so they keep your cells together. And then finally, there are also proteins that are attached to your cytoskeleton. And we'll talk about your cytoskeleton later in the semester. But cytoskeleton is very much like your own muscular skeletal system. It gives your cells structure. But again, we'll talk about the cytoskeleton later in the semester. So there are typically 6 broad classifications of proteins that can embed themselves in your cellular membrane. And depending on their shape, they have different functions, right? So you can see that a transporter has a very different shape than an enzyme, which in turn has a very different shape than a cell-to-cell adhesion protein. So you can see pretty clearly that the structure of these proteins dictates what they do, because a transporter could not be involved in cell-to-cell adhesion, the same way a cell-to-cell adhesion protein could not be involved in enzymatic reactions. So again, structure and function is very, very strongly linked in our proteins. And again, I've mentioned this before, but remember this idea. If you remember nothing from this class, remember that structure relates to function. The structure of proteins, the structure of carbohydrates, the structure of lipids, all relates to their function. If you change that structure, you change the function. Remember that. The most important thing I will teach you in this lecture is structure is tied to function. And as we dive into proteins later in the semester, you'll see why that's the case. But just keep that in mind. Structure relates to function. And again, just by looking at this diagram here, it's easy to see this could not do the job of this, or this do the job of this. Again, because it's all they have different structures, and that structure dictates the function. Um, and in terms of our proteins, there are, as we mentioned, there are anchoring molecules here. And our anchoring molecules are modified lipids. And these anchoring molecules are sort of a small class of things we're just going to just mention, but they're involved with anchoring things to one another. And they do this with chemical bondings, and is typically linking nonpolar regions, inserting themselves into the lipid bilayer itself. So again, just as a note, there are special types of anchoring molecules inside your membranes, but again, not too important for the purposes of this class, but just wanted to make mention of them. You might see them in your textbook or something like that. And they're just designed to anchor things, as their name implies. Now, our membrane proteins they fall into a couple of classes. So our first classes are integral proteins, and these are ones that span the lipid bilayer. They're also called transmembrane proteins. Now, the nonpolar regions of the proteins, so the ones that are going to be hydrophobic, are embedded inside the interior of the bilayer. And then the polar regions, which are going to be hydrophilic, are going to be present in the cytoplasm or outside of the cellular membrane, which is kind of interesting. And I'll show you what that means right now. So in this case, we have non-polar ones that are embedded inside the phospholipid bilayer, again, because they're hydrophobic, meaning they hate water, very much like these fatty acid chains. So there's no water inside here. But our hydrophilic ones or polar ones are going to be outside here in, say, the water, or inside the cytoplasmic space here. But they have multiple domains. So there's what we call a transmembrane domain, which causes them to span the lipid bilayer itself. So the transmembrane domains, we have inside the membrane, and then we have connectors on the outside. They're not beta sheets or like anything like that, but they're just sort of connector pieces of proteins here. But they do need just a single single transmembrane domain. So as we see here in B, they just need to be able to cross the membrane once to be considered transmembrane, or they can cross it multiple times as we see here in A. But anything that's transmembrane has to fully span, connecting the outside of the cell to the inside of the cell. Next up on our train is going to be our pores, and our pores are are comprised of extensive nonpolar regions within a transmembrane protein that create a pore through the membrane itself. So think about it like a hole in Swiss cheese, right? You have just this hole that goes through the membrane. And you can imagine these pores are designed for transporting things across the membrane. They could be very large things or they can be very small things. It just depends. Now, pores are made out of what we call cylinders of beta sheets, and these are called beta barrels, which is aptly named because you'll see what they look like in just a second. And I'll just show you what they look like. So we're all well, actually, I don't have a good. Sorry, I thought I had a better picture. But these pores again, they look like barrels. So think of like the like an empty pen, and that's sort of what a beta barrel looks like. It's just a pore, just a hole. You can think of like if you took a piece of paper and you stabbed a hole with it with your pencil, that's what a pore looks like. Really, really sort of straightforward structure. But pores are involved in terms of transmembrane proteins. They're involved with moving things across the cellular membrane. Because remember, the cellular membrane has to be fluid. It has to move things from inside the cell to outside of the cell, and from outside of the cell to the inside of the cell. And it does this through two mechanisms. So the first one is what we call passive transport. And passive transport is the movement of molecules through the membrane where you don't require any energy. So you don't physically have to expend any energy to move something across the membrane. But they are moving via what we call a concentration gradient, and they're moving via diffusion. And just to show you what diffusion looks like, diffusion is the movement of molecules from a high concentration to a low concentration. So what ends up happening in this example up here, we have a high concentration on the left here. As enough time passes, diffusion as a process moves molecules to the other side of this barrier here, creating equal concentrations on both sides. And that's how things move in your cells. Well, some things move in your cells. They can move, say you had a very high concentration of sugar outside of your cell. Well, diffusion will allow the sugar to move inside of your cell, again, going from high to low, until there's equal concentrations of sugar on both inside or outside of your cell. And if you want to visualize this with a sort of a more real world example, drop food coloring into a beaker of water. Eventually, diffusion will spread that food coloring throughout the liquid of your liquid here until the entire liquid you're looking at is stained red with your food coloring. So that's diffusion. Um, but that's diffusion. And so diffusion relies again on gradients, but the membrane itself can be actually a relatively large barrier to itself. And because as you cross it, you're crossing through something that is first hydrophilic and then hydrophobic and then back through something hydrophilic. So certain types of molecules have a relatively difficult time moving across. And so molecules that are nonpolar will move across the membrane freely via diffusion until the concentration is equal on both sides. However, molecules that are very small but they're polar will move slightly, but they won't move until there's equal concentrations of both sides. Because they're polar, they find it very hard to make it through your cellular membranes. And as you get very, very large, so as you start talking about sugars or very, very large other types of polar molecules, or potentially ions, it's very hard for those things to move across the cellular membrane. They have very, very limited permeability across your membranes. Certain things have trouble moving across your membranes. We need to talk about the other type of transport across your membranes, which is called facilitated diffusion. Now, facilitated diffusion essentially targets molecules that cannot physically cross the membrane. So they're charged, they're polar, or they're very, very large. They can't move, and when I say they can't move across, they can't move through the cellular membrane itself, right? They can't move through that phospholipid bilayer. However, we can embed certain classes of proteins to move them actively across them. And these include channel proteins as well as channel proteins and carrier proteins. Sorry, I don't know why that was hard to say. But again, remember the membrane itself is selectively permeable. And so there are a couple different classes of channel proteins. So the first one is ion channels, and these allow the passages of ions, so either positively charged or negatively charged atoms, through your plasma membrane. And so these are what we call gated channels, and they open and close typically in response to a stimulus. And so an ion channel, just to give you an example of like sort of your real world life, the way your muscles contract is the movement through ion channels. So you move ions in your cell and then out of your cell to contract and draw in your muscles. This is true for speaking. This is true for lifting weights or anything, picking up something, right? So that's what our ion channels are. And in terms of how we move things with our ion channels, there are three conditions that determine the direction. And these include the relative concentration on either side of the membrane, the voltage differences across the membrane, so how much charge you have on one side versus the other, and whether the channel itself is open or closed. Because as I mentioned, ions don't diffuse across the membrane. They just don't have the capacity to do that. So they rely totally on these channel proteins, these ion channels. And again, sometimes these channels will be closed, which will not ions across. But also the concentrations on one side of the membrane influence whether the channel will actually open, as well as the voltage differences. And what do I mean by voltage differences? Just to sort of show you, say if I'm transporting, say, calcium ions across, if if I have a lot of calcium on one side, my voltage is high because calcium is a charged molecule, and that's higher on this side versus on that side. But the idea being is we use facilitated diffusion in these form of these channels to move ions from outside of your cell to inside of your cell. And this might not seem like an important thing right now, but keep that in the back of your mind because we're going to talk about movement of things across the membrane a lot when we start to discuss metabolism and things like that. But the movement of ions is a really important thing that your cells can do because ions are involved in a lot of different cellular processes. And so as I mentioned, the other type of facilitated diffusion proteins are called carrier proteins. And these themselves help transport ions and other things that are dissolved. But in particular, they're looking mostly at sugars and amino acids. Now, our carrier proteins do rely on diffusion to move things across, but it does require a concentration gradient to occur. And ultimately, they must bind to the molecule that they have to transport until they get saturated. And then they move them across the membrane. So let me show you what I mean by that. So we have carrier proteins. We have some sort of compound out there. It could be a sugar, it could be amino acid. The amino acid binds carrier protein, and then what it does once the amino acid or sugar binds, it causes the protein to change shape, and then it essentially expels whatever is attached to it into the cytoplasm of the cell. And then once it expelled things, it goes back to its original shape, which is sort of this weird sort of shape. But again, it binds, changes its shape again, pushing the molecule through. So this is facilitated fusion. And now this won't happen unless there's a or purple molecules on the outside versus the inside to move things from outside to inside, as well as vice versa. And so that's how we move molecules. And sort of one of the last things we need to talk about for how we move things across the membrane is osmosis. And it's not this sweet movie, rock from the 90s or 2000s, Osmosis Jones. It's simply referring to how water moves inside your cell and outside your cell. So everything we've talked about with diffusion is how everything in the world but water moves, because water has its own special process and water moves via osmosis. Now, as I mentioned, your cells are basically just big bags of water. And they basically, not only are they big bags of water, they sit inside an even larger bag of water. And so water moves in and out of your cell via this process of osmosis. And what this is, it's like diffusion, where water, but it's not how a chemical moves, it's just simply how water moves. And water moves from a concentration of a solute that's very low to a concentration that's very high. So let me show you what this means. So we have water in this osmosis, what we call a U-tube, not like, YouTube as in like Google's thing, but just a tube that's shaped like a U. I guess that's kind of a pun. But what you see on this molecule, in this tube, is we have water and we have urea dissolved in this water. And what you'll notice is that there's some membrane here, and there's one molecule of urea on this side, and there's 1, 2, 3, 4, 5 molecules of urea on this side. So we have a concentration gradient of urea, where we have more urea on this side and less urea on this side. So what osmosis tells us is that water will move from this side, where urea is in low concentration, all the way into this side, where urea is in much higher concentrations. Now, I know that seems kind of weird, right? Because diffusion should tell you Water should move so that there's equal concentrations of water on both sides. But that's just not the way water moves. Water moves from low concentrations to high concentrations. And what ends up happening is as I move water from this side of the U tube to this side of the U tube, it actually ends up creating a system where urea ends up being in the same concentration on this side and the same concentration on this side. But it does it by increasing the volume of this side. I know it's a weird thing to think about, but again, all water is doing, it's moving from low concentrations of whatever is dissolved in it to high concentrations of whatever is dissolved in it. Again, weird, but that's just the way water does it. Now, there is some terminology surrounding osmosis. So when two solutions have different osmotic concentrations or different concentrations of solutes, we have what's called a hypertonic solution, where there's a very high concentration of a solute. And we have what's called a hypotonic solution, which is we have very low concentrations of a solute. And again, a solute is anything dissolved in water. It could be sugar, it could be salt, it could be urea, it could be anything. Now, when solutions have the same exact osmotic concentrations, we call them isotonic. So if I have two solutions, they both have 50 molecules of sugar, well, they're isotonic because they have the same osmotic concentration. And then you might think, well, this doesn't have quite, why do I care about this? Well, it does have some real world implications. So of how water moves across cells and some other sort of other aspects of this. So we can look at an isotonic solution where there's equal concentration of a solute inside and outside of the cell, which means there's no net movement of water between inside of the cell and outside the cell. Next, we can look at a hypotonic solution where there is a higher concentration of solute inside the cell versus outside the cell, which leads to water rushing from outside the cell inside of your cell. Or we can think of a hypertonic solution where we have higher concentrations outside of your cell versus inside of your cell, causing your water to rush out of your cells. Now you might think, okay, cool, whatever. Well, this actually has some real world consequences. So in an animal cell, such as a red blood cell, in a hypotonic solution where you have higher concentrations outside the cell versus inside the cell versus outside the cell, it causes water to rush into your cells, causing them to rupture. So anytime red blood cells rupture due to osmotic pressure, it's because water's rushing in, causing them to essentially explode. In an isotonic solution, and again, that's equal balance, so there's no net movement of water inside or outside your cell. And then finally, we can have what's called a hypertonic solution, where there's high concentrations outside the cell and low concentrations inside, causing water to rush out of your cells, causing them to dry out, to shrivel up and die, which is not fun for your cells, right? The only time your cells are happy is when they're in an isotonic solution. Now, that being said, plants themselves do take advantage of hypotonic solutions. I will make a note. So plants have a very special compartment called a vacuole, and we'll talk about this as we talk about cellular structures soon. But this is basically a storage a container for water. And so plants take advantage of hypotonic solutions to inflate their vacuole to give them what's called turgor pressure. And for those of you that have ever gardened, have ever grown any food, if you've ever seen your plant wilt, one of the reasons your plants are wilting is because they don't have enough turgor pressure, because they don't have enough water or they don't have enough dissolved solutes to give them a hypotonic solution to keep them rigid. Because turgor pressure in plants, again in hypotonic solutions, makes them rigid, which makes plants stand up. So if you ever have a wilted plant, the first thing you should always try to do is try to water it, because usually it means it just doesn't have enough water present. So for those of you that have no luck growing plants, potentially like my wife does, one of the reasons might be you just don't water it. And again, it's all due to the presence and absence of water inside the cell. So we've talked about diffusion, facilitated as well as passive diffusion. We're going to talk about a more active way of pulling things that you need inside your cell versus outside the cell. And this is what we call active transport. So the other processes we've talked about require no energy. They rely on concentration gradients to move things, whether it's water, whether it's molecules, whether it's anything. But active transport physically requires the use of energy. And as I mentioned in the previous part of this lecture, the energy currency of a cell is going to be adenosine triphosphate, or ATP. And active transport requires ATP to move substances either from high to low concentrations or low to concentrations. Typically, moving when we're doing active transport, we're typically moving things from low concentrations to high concentrations. And again, it's moving things from inside the cell to outside the cell or outside the cell to inside the cell. But again, it all requires some form of energy to do it. Now, there are many different types of active transport proteins that are embedded inside your cellular membrane. And these are called uniporters, which move one molecule at a time, symporters, which move two molecules in the same direction, and antiporters, which move two molecules in opposite directions. And we're going to talk about a very specific type of transporter here in terms of active transport. And we're actually going to talk about the sodium-potassium pump because it's a particularly important pump for your own biology, just as a note. But as all active transports, they use ATP and the antiporter. So moving, what they're doing is moving one molecule across one way and another molecule the exact opposite way. But the sodium potassium pump pumps three potassium ions outside of your cell while bringing two sodium molecules out of your cell while bringing in two potassium molecules into your cell. And the movement of them is typically in a concentration gradient. So typically speaking, you have more sodium inside your cell versus outside the cell and vice versa for the potassium. But to do this, you use ATP to move it. And what ATP is actually doing is actually just changing the shape of the carrier protein itself, this antiporter. And so let's look at what this looks like. So we're trying to move sodium and potassium. So in this example, potassium is going to be little yellow balls, and sodium is our little purple squares. Now we're trying to move potassium and sodium in or out of the cell. Just as a note, if you want to read about this pump, I linked a very nice Wikipedia article down here that discusses how this moves, but we'll just talk about basically how it works. Now, the idea being we have ATP and 3 sodium ions will bind to this pump. Now, once the idea being is once the ATP binds, it transfers the phosphate group to the pump, thus transferring energy to the pump. Now, once you transfer energy to the pump, what this does is it causes the pump to change shape. So it looks like like this shape here. Once you add the energy, it changes shape to be like this. So what it does is it expels the sodium from inside your cell to the outside of your cell. Then what it does is it allows potassium to bind to the other side. Once the potassium binds, it causes the phosphate group that we tacked on up here to disassociate it. And once that phosphate group disassociates, it causes the protein to go from this shape back to its original shape. And this can be repeated over and over again to move potassium and sodium across your membranes. So it's a really handy antiporter, right? You're exchanging ions from another, right? And these ions physically can't diffuse across your membrane, right? Because they're charged, and charged things can't move naturally across your membrane. They need help. And in this case, we're using sodium potassium pumps to move them. And again, it does require some energy, and that energy for your currency for your cell, again, is always going to be ATP or adenosine triphosphate. Now, we can do also what's called coupled transport, which uses ATP, but it uses it not directly like we saw, but indirectly. And it uses energy released when a molecule moves by diffusion to supply energy to an active transport of a different molecule. And this is what we call a symporter. And the one we're going to talk about is the glucose sodium symporter. It's a really handy thing. Now, this symporter captures energy from the diffusion of sodium across the membrane to move glucose against a concentration gradient. So let's talk about what it looks like. So in this case, we have glucose, which is our sort of ring structure here in green, and we have potassium and sodium here again. So we're still looking at that potassium-sodium pump. Now, we have two different pumps here, right? We have our potassium sodium pump, which is an antiporter, and then we have a symporter here, right? Antiporters move things in both directions. Symporters move two things in the same direction. Now, we couple these two together. So we talked potassium and sodium pump already, right? We move sodium um, from inside the cell to outside of the cell to potassium. But then what we do is as we move enough sodium across, as you'll notice, we have more sodium outside the cell than inside the cell. Well, what we can do is use that gradient that we've developed, right? More sodium on the outside versus the inside to help transport glucose across the membrane. So as we get enough sodium outside, it will bind to this protein and allow glucose outside of your cell to bind to the protein, thus bringing glucose across with it. But as you'll notice, we're going against the concentration gradient here for glucose, right? We start with a lot of glucose inside the cell and very little glucose outside your cell. And so you can imagine this type of transporter is important for moving food sources, right? If I'm trying to move sugars like glucose, it's a really handy way to do it because normally speaking, if I wanted this glucose to go from outside my cell to the inside of my cell, I would have to rely on a gradient of glucose. But by using energy, I can capture glucose by moving it in a concentration gradient with sodium, even though glucose itself is moving against the concentration gradient of glucose. So it's a pretty handy little technique of how we move things with active transport. There is other types of active transport that we should talk about, and these are bulk transporters. There's a first one which is called endocytosis, which is of movements of substances into the cell. And there are two types of endocytosis that we'll talk about later in the semester. The first one is phagocytosis, where you bulk, essentially engulf something. So this is what your white blood cells do. They basically like gulp onto it like Pac-Man. They just kind of bulk, just nom it up. Can't think of a better way to describe it. And then we have what's called penocytosis, which is the direct uptaking of large amounts of liquids. Again, phagocytosis is the engulfment of matter, solid matter, particular. Penocytosis is directly in liquids. And then we have what's called exocytosis, which is the opposite, where we bulk move substances out of the cell. And we'll talk about exocytosis in a couple classes as we talk about how our organelles are linked together and things like that. But just to sort of show you what endocytosis looks like, we have phagocytosis of a bacterial cell. So this would be akin to your white blood cell. So you have the plasma membrane, and what endocytosis does, or in this case phagocytosis, it's just basically your cell membrane extends out or extends in, allowing the bacteria or whatever large substance you're trying to do come into your cell and then be engulfed by your plasma membrane. And by being engulfed in your plasma membrane, you basically cordon off the bacteria in its own, what we call a vesicle. And you keep the bacteria inside you, and if you're a white blood cell, you eventually kill this bacteria. Now, you can imagine you could do this in other ways as well, right? Maybe there's a big food source here and you want to engulf that food source as well. But it's anything particulate. But the example I have here for you is how your white blood cells work. And it's a really cool sort of way to look at it because you can see it here in this electron microscope where you have the engulfment or phagocytosis of this bacteria by your white blood cells. And again, penocytosis is bulk engulfment of of small solutes that are dissolved in water. And it's the exact same way. Your membrane buckles, it engulfs whatever you're trying to do, and it cords them off. And what we have here, you can see little tiny, again, these are called vesicles of whatever we have here. You can do this for a number of different things. So there's a lot of organisms that will do toxins this way. So they'll engulf toxins and to keep them inside of their own little special container to essentially prevent them from being so toxic. Now, the other type of bulk transport is exocytosis, which is the discharge of materials outside of the cell. So if you have something inside your cell that you want to get rid of, you put it inside a piece of a membrane or a vesicle. And that vesicle gets transported to your membrane, it fuses to the membrane, and then eventually the membrane breaks open, spilling whatever you're trying to do into the environment. Now, this can be done to do a couple different ways. So this can be done to excrete waste from your cells. So what your cells do is they do an exocytosis to excrete waste into your bloodstream to then be sent to your kidneys and your liver and your colon for excretion from your body as a whole. But this is also the way we secrete hormones, so testosterone, estrogen, and so on and so forth. This is also how your neurotransmitters work. So they use exocytosis to dump neurotransmitters such as serotonin, dopamine, and so on and so forth between your neurotransmitters to make you feel happy or sad or excited or any of the other emotions humans feel. And you can also use it for excretion of digestive enzymes. So in your stomach, your stomach cells use exocytosis to excrete digestive enzymes into your stomach and into your small intestine to digest your food. So lots and lots of uses for exocytosis in terms of functionality of your body. So that's going to sort of wrap up today's class. I thought this was going to take us slightly longer, which is why I didn't have an exercise for us, but I think it's okay to end a little bit early today because we did cover a lot of ground today. We started off with macromolecules and we ended on talking about movement and membranes and stuff like that. So we did cover a lot of ground today. So I think it's okay to end class a little bit early with no exercise for the day. So as I mentioned, that will be the end of class today. If you have any questions about what we talked about today, please let me know. I'll hang around for a little bit, or you can obviously email me or schedule a time to meet with me and discuss either in person or on the internet to discuss these topics. But don't forget to do your lab. As a note, if if you're having trouble with Labster, you do need to make sure you create an account. And if you're still having trouble, you really need to get on the phone and talk with the folks at Labster. Because unfortunately, I just don't have the capacity to help you with technology issues with Labster. I don't have access to all the things Labster is. So if you are having problems, again, please let me know. But please do get on the phone and talk to Labster. So with that, I will see you all next week, same time, and obviously same place because we always meet on Blackboard Connect. But if you have any questions, as I said, I will hang around. And I saw a lot of people were dropping in and out of the session. I will post this lecture, the recording of it, to the weekly folder for you so that you have, in case you want to go back and look at something that you didn't quite get because you kept dropping out or something. So with that, I will see you guys next week. Take care.
Hi, I have just one more thing to say about the homework too. You told that the answer should be there. I already look on this and I can see the answers, but not which one is the right one or which one I mark it. So just to let you know that maybe something is not working
Um,
there, but that's
let me
fine. So
hold on. Let me just go back and look at the settings. One second, because it should show you right away. But let me see maybe some weird setting. Um, so when you're going back to look at the homework, how exactly you're trying to access it?
So I go to homework two on the grade one. I click on this, like say see attempts, and then I can see the answer and the question and the answers, but I cannot see which one is the right one, which one I mark it. I can just see like questions and answers.
Interesting. Okay. Hold on a sec. Let me do this. Let me make it available again. Maybe that will work. So why don't you try refreshing your page and then I changed some settings. Maybe that will help. After submit all answers. Ah, I get it. Hold on a sec. Hold on a sec. Give me one sec. I found the setting that needs to be clicked. All right. Now refresh your page and take a peek at it. See what you see. That should have fixed it.
Yeah, now I'm able to see which one is right and wrong.
There you go. All right.
Thank you.
Perfect.
Bye,
Yeah.
have a great day.
You too. All right. Perfect. The date for the first exam is on. If you go to the syllabus, it's there. I actually think it's coming up, if I'm not mistaken. I can look at the syllabus to let you know right now. Whenever the internet here at school decides to work. Not try just decides to learn. Come on, come on. What's going on, internet? Why are you taking so long to load? The just as a note, the exams are all online, so they are open note, open book. What was your question, Catherine?
I have two questions. Okay, so the first one is, does the lab homework and the extra credit discussion board are due Sunday by midnight?
If I'm not mistaken, so the homework is due October 4th by midnight,
Okay.
and the extra credit discussion board, let me just double check. I don't want to tell you the wrong thing here, but I can tell you exactly when it's due just by looking at the assignment. Come on, what is wrong with the internet at school today? Sorry, the internet's just being very, very slow for some reason. Yeah, so it's, yeah, so when I look at it on my end, it's the 4th.
The discussion board?
Yep.
Okay.
October 4th, that's when it's available. So
And then the lab 2, right?
lab 2 you have till Friday to do.
So yeah, about the lab, I tried like to do it yesterday by searching like by the name that it says like introducing food to molecules, I think, but it doesn't show with the name.
So why don't you log on to Lapster and share your screen with me so I can see what your problem is and maybe I can
Okay.
help you. So
Okay.
head on over to Lapster, and then at the bottom of the screen there's like a little thing that says collaborate panel. It's purple. If you go over to the third option, it's a little arrow that says share content, and you can share your screen with me.
Let me see. Okay, wait. You said at the end?
So at the bottom, so in the right hand corner, there's
Okay.
if the right hand menu is not open, there's a little purple arrow that says open collaborate panel in the bottom right hand corner. You would click that. If that panel's not open, but if it is open, you go down to the bottom. So the first one says chat. It's a little text bubble. Then it says attendees, which is 2 people, and then it has the final one, just share content, which is a little arrow. So you can click
Okay.
the little arrow one on the bottom, and then there's an option that says share application / screen, and you can click that and share your screen with me so we can look at it
Okay,
together.
I'm going to find the little box. No, it's not here. Let me see. No, this is the blue. Says hi there or whatever. No, it's not this one. Let me see. Oh my god. So the only thing that I can see is a thing that it says like in the top it says all biology, chemistry, physics, logout, help, and dashboard, and then at the end of the like if I scroll down all the way to the end, it says the lab of biology that it says your diet and your DNA, and then at the right, like in the bottom right, it says like a message symbol, and then I click there, but it says like hi there, we're live from Monday to Friday a.m. to 1 a.m. So yeah, I don't know.
So this is what you should see when you click the link for Labster on Blackboard. This is what you should see. This is the link. So when you go to Blackboard, just as a note, you go to Labster, right? You go to lab materials here.
Yeah.
Come on. The internet's slow today. Sorry.
You're fine.
It's just like the most frustrating thing. And so you can click class Labster link, and that will take you to the space. Or if you're, you want to go to the week of
I always
today,
go
you just
like
click
to the week.
this for this week.
Yeah.
Mhm.
But when I click it, it just like appears all the labs, and I'm trying like to find the one that says Introduction to Food Macromolecules, but I can't find it. Even when I search it in the type it in the search box.
So this is what you should bring you to Labster, which is what I had open already.
Yeah.
Is on this page is just scroll down. So if you just Control F and type food, and provided it can search. Come on.
It's a little
This is
bit
such garbage.
blurry, but I can see. Let me see.
Sorry, this is absurd.
You're
It shouldn't
fine.
take this long. So you can see it right here.
Okay, let me see when, because it's a little bit blurry. Okay. Oh yeah, it's just the first one. Let me see here if I can. Oh, so is Control C food. Oh yeah, yeah.
All right, sorry, I got kicked out of Blackboard.
No, you're fine. I think I found it. Yeah.
Yeah. So just to sort of show you, like this is where the link on. Oh, hold on a sec. Wait till this comes back. So this is where the link on Blackboard takes you, and if you scroll down through it,
Mhm.
you'll eventually find what you need. You don't need to search through the thing. You'll eventually find it. So, um, you know, where was I just saw it. Introduction to food macromolecules right here.
Yeah.
Then you click it and you do it.
Okay. Thank you so much for your help.
No problem.
Nice day.
You too. I'll see you next week. Are you just hanging out, or do you have a question, or? Okay, I'm going. Bye-bye.